But maybe let's start now. So it's my pleasure to introduce Elze Hesse, who's going to be giving our second state-of-the-art talk this year. So Elze works on the ecology and evolution of cooperation and exploitation of the microbial system. She actually started with a PhD on the ecology and evolution of plants, and that was in Freiburg in Switzerland. Actually, she continued working on plants for a little while. She can tell us a little bit more about that. And went afterwards to do a fellowship, a research fellowship at the Swiss National Science Center Foundation at the, it was a fellowship funded by the Swiss National Science Foundation.
Yeah, to go to Oxford.
To go to Oxford.
Yeah. They pay for foreigners to go abroad, which is amazing.
Actually, they pay really well as well. And then you did a postdoc at the University of Leiden in the Netherlands before coming here on our campus, again as a fellow in 2013, was it? A little while ago. Yes,
I think it was. It's all a bit of a blur. But yeah.
And then Elze got a really prestigious UKRI Future Leaders Fellow. Oh my, today is not my day. In 2021, which is going to be for 5 years. 4. Okay, so I'm not going to get it.
3 potentially, but yes.
Okay, perfect. And today you're going to be talking about living soils, the role of microbes in soil health. Welcome.
Thanks for the introduction, Camille, and thanks for reminding me how long I've been here already. It's been a while. So for the people who don't really know what I'm doing here, just going to give a brief background how I actually got here and about my scientific background. So as Camille said, actually before coming here, I've mainly worked with plants. So I did my master's already on plants, looking at the effect of logging on liana diversity. And then I moved on to Switzerland to do my PhD and to look at the optimal evolution of optimal flowering times. And this was in a clonal invasive weed that occurs in the pastures and it is toxic to cattle. And then I moved to Oxford to actually look at the evolution of sexual systems in plants. So I worked with this kind of small weedy species, Mercurialis annua, and looked at the evolution of dioecious in plants and the role of interpollination in the evolution of dioecious. This plant occurs in really fragmented habitats, and as such is a really good model system to see under what conditions dioecious breaks monoecious, so where you've got both males and females flowers on a plant, when does that break down and how does that actually affect the stability of populations? Then I moved to Leiden to actually work on oilseed rape and to actually look at if we can predict when under what conditions oilseed rape becomes feral, so when it is able to escape from the pastures. And we focused on one specific trait, which is glucosinolates and the anti-herbivory chemicals. And we actually found is that if you've got more of these chemicals, it helps you on early on in life, but actually that difference disappears later on in life. So in between this, it looks quite a smooth research trajectory. Sorry, I found that really hard to pronounce. I did some consultancy work. So I was a botanical consultant for BHP Billington in Guinea, in Africa, to do an environmental risk assessment before they actually start digging for iron. So we collected lots of plant material and also visited local villages to actually see what the people there actually thought about the opening of the mine, and a lot of the field assistants were actually from the local villages. So a brief summary is interested in evolutionary ecology, views plant systems, and then while actually doing a lot of work in the field, and it was mainly field work, I started becoming fascinated not only what happens actually above ground, but also below ground. So what is the role of soil microbial communities in actually determining ecosystem health and plant health, and whether we can actually use those communities for our own benefit. So when moving here in 2013, Angus Buckling had a postdoctoral research fellowship available, which I applied for, and I got that one. So I started looking at microbial communities, and you might think, why microbes? So just like higher order organisms, microbes display a lot of social behaviors. They can help each other in scavenging food, but they also kill each other quite often by producing toxins. So these, as you can see here in some examples, so some of these colonies don't mix, so they inhibit each other's growth. In the top bit, you can see Dictyostelium, and you can see Myxococcus in the lower picture, which is taken by Mikvos. And these microbes, they cooperate by actually producing a fruiting body when resource conditions are poor. And by doing so, some of these individuals will end up in this little stalk here, and they don't produce any spores, so they actually don't contribute to the next generation. So this opens up the question, so why do you want to cooperate with other individuals within these communities? So species interactions in microbial communities are diverse, like I said, so they can be mutualistic, where both of the interacting species actually benefit each other. They can be exploitative, where only one of the species benefits, and quite often they're competitive. And that's been recently reviewed by Palmer and Foster, and they found when they actually looked at communities that were collected in the wild, or actually where they took samples from the wild, put them in the lab, and they measured interactions. And they found for soil bacteria, for example, that most of the interactions are negative, meaning that they are either exploitative or competitive. So it's really important to know the nature of species interactions in communities, because it really determines combined community functioning. So there's a lot of theory out there that shows that if you've got more mutualistic interactions in a community, this might enhance productivity, but on the downside, it might also destabilize communities in the face of environmental change, because if one of the interacting species falls away, then it might lead to extinction cascades. So a lot of these microbial interactions are driven by public goods. So public goods or services are actually compounds or functions that not only benefit the producer, but also individuals in the wider community. You can see that here. There's some bacillus or streptomyces colonies that produce antibiotics, and in doing so, sometimes this actually benefits other community members that don't produce these antibiotics, and in that way they actually help others to survive. Another example is the production of siderophores, which is the model system I'm working on. So in that case, you've got a producer cell, so these pink little things are siderophores. They bind to iron. So iron is quite often available in a non-soluble form in the environment. So bacteria cannot readily access these, so they produce these siderophores to mobilize iron, and then it's taken up by a matching receptor, and in that way it can actually benefit a non-producing cell. So community public goods, they benefit others, and quite often these dynamics have been studied within species. So we know a lot of public good dynamics from studies in labs. So for example, the evolution of siderophore production in Pseudomonas aeruginosa, which is a well-known pathogen. But we have actually no idea what actually happens at a community level. So if you actually increase community complexity, do we still expect the same rules and patterns to hold compared to what we find within single species? And this is quite important, because if you look at a community and you've got like these dynamics playing out, so you've got cooperatives that actually produce public goods, but you also have actually strains and species that exploit the public goods. So what are the community-wide consequences of that? So that's been captured by the Black Queen hypothesis. So what that states is that if non-producers are able to invade a microbial community, then this actually reduces community-wide public goods production. And also what they say, it leads to a race of the bottom, where actually one of the species is actually stuck as a helper, while the other ones are actually exploiting or benefiting from the presence of the helper. It also leads to the loss of leaky traits and genomic streamlining. So as a consequence of being able to use a public good produced by others, this could lead to loss of function mutations and genome size reductions. So why do we want to know the dynamics of public goods in communities? So it's important to know these because they really underpin community functioning. For example, they're important in detoxification of metal-contaminated soils, which I will tell you more about today. But they're also involved in biofilm formation, because usually when there is an infection or when you grow species on a plate and you want to look at more at the mechanisms of the infection, you find that not all of the species pay their fair share. So they actually benefit from the public goods, the biofilm produced by others. And there's other examples where actually these community functions are really important and driven by how different species actually might help or exploit public goods. So that's basically the main aim of my research, is to actually understand when and why, under what conditions, different species cooperate. So when you look at cooperation within species, there's been a well-developed theoretical framework. So we know that kin selection is really important. So usually the production of these public goods is costly to the individual. So then why would you want to help others in the environment? And one reason this could be is because it not only benefits you as a producer, but also related kin. So in that way, you've got indirect fitness benefits coming back to you. But this obviously is not the case for different species that are quite often not really related to one another. So there's only direct benefits of social interaction between individuals when you talk about social interactions. So, like I already told you a little bit, so I'm going to look at bacterial siderophores as a key social trait. So this is a good social trait to look at, these community-wide interactions, because they have both benefits within species, but also across different species. So we can actually at the same time look at the importance of kin selection, but also between species selection in determining the community-wide production of public goods. So in terms of within species cheating and cooperation, we can look at siderophores in the context of ion acquisition. So like I said, iron is often available in non-soluble form. So species have evolved the ability to produce siderophores that make these metals or iron more mobile. But it can only be taken up by a non-producer cell if it's got a matching receptor. And this is often quite often limited to within species interactions. There's some examples that there is exploitation of siderophores of other species, but mainly it is within species. However, in the context of metal detoxification, the siderophore binds to a toxic metal, and because this complex is too big, it can't diffuse back into the bacterial cell. So detoxification takes place outside of the bacterial cell, and because you don't need any matching receptors, this would benefit potentially anyone living in the near vicinity of the producer cell. So we've got these two different roles of bacterial siderophores. And when I started my PhD, it was known that actually non-producers and producers actually differ in their metal resistance. So it could be shown for Pseudomonas aeruginosa. If you've got a siderophore producer and an isogenic knockout mutant, that the mutant doesn't cope very well with metal stress, meaning that siderophores are important to detoxify the environment. What wasn't known was whether there could be exploitation of these siderophores in terms of metal detoxification, or whether you could actually share these siderophores between species. So what I first did, together with Siobhan O'Brien and others, was actually look at in the lab on very controlled conditions. We had copper polluted microcosms, and we had like a strain of Pseudomonas aeruginosa, PaO1, that was able to produce pyoferdin, the main siderophore, and a knockout strain that wasn't able to produce any siderophores. And what we tested is if we added like pure siderophore isolated from a different species, could this rescue the growth of the non-producer strain? And this would then be a proof of concept showing that actually siderophores can be shared between species. We carried out a similar experiment under more complex conditions in compost, where we had the same two strains that were either grown in isolation or both together, and in the presence and absence of the microbial community, with the idea that members of the microbial community might be able to actually detoxify copper in this case, And in doing so, it would benefit the non-producing species. So what we found is, it's exactly what we found, which is always nice. So here on the left, you can see if we only have the copper, there's no detoxifying siderophores or community members, then the non-producing basically cheat, or not, however you want to call it, does not fare very well compared to the wild type that does produce the main siderophore pyoverdin. However, as soon as you add foreign siderophores, so we tested about 4 or 5 different pure ones, or the community members, then this difference disappears. And actually, at one point, the non-producer has a selective benefit because it actually does not bear the cost of producing siderophores itself. So that was really nice. So within the model species, for which we already knew a lot, we could actually confirm that siderophores are important in toxification, but that it also can be shared across different species. However, this is a very high siderophore producer, and still don't know much about siderophore production in natural community members. So from this kind of experiment, we isolated 10 different bacterial isolates that differed in their colony morphology. So they were like yellow, pink, all sorts of colors, which made me very happy. And we did pairwise competitions between those in unpolluted and copper polluted compost. And not only did they differ in their colony morphology, they also varied a lot in their siderophore production. So we wanted to test the idea that on the copper polluted conditions, the strains that were sensitive to copper, that did not produce siderophores, could benefit from the presence of another species, with the result that its growth could be rescued. So this is, for example, the producer in copper. This is the non-producing strain. And then when you combine them together, the orange one, the non-producer, would benefit from the presence of the other species. Well, we would not expect that to happen actually in the unpolluted compost, because under these conditions, strains will compete for iron and the siderophores cannot be shared across the different species. So what we expected is that there will be more negative interactions in compost compared to copper polluted conditions. And that is basically a main hypothesis in ecology called the stress gradient hypothesis, which predicts these interactions to happen, stress shifting the balance towards more positive interactions. So like I said we had like 10 common compost isolates that varied in their siderophore production, and they also varied a lot in what they looked like when plated on agar plates. So we grew these species in isolation, but also together at a 1:1 ratio in copper polluted compost. And then looked at the comparative versus permaculture.
Oh no, I did something I shouldn't have done.
Wait,
Yeah, I think they're all muted now. No.
I think you need to, if you go back to the PowerPoint.
They've just muted. Okay.
Just go back to my PowerPoint.
Oh God, it's not easy.
Hey, Dan.
Okay, I'll go here. There you go. Perfect.
Yes. So okay. Yeah. So what we first. Oh no, this thing doesn't work.
Just need to click into it once.
Okay. Doesn't work at all anymore.
Sorry. Do you want me to just?
No idea.
Should we go exit, show and start again?
I think if you just move the cursor over to the main presentation.
Yeah.
Oh yeah. Okay.
Yeah. Okay.
There it is.
Yeah.
Yeah. Now if you click into that.
No.
No.
Wait, I can actually wait. Yeah.
Do you want me to? Oh.
I'll see if this wait. No, it doesn't work. Okay, I'll just go to this slide. Yeah. So what we first wanted to confirm again, that actually siderophores, they do provide protective benefits in copper polluted compost. And that's, first of all, we actually verified whether the copper was actually at a toxic level, and you can see that densities are of the 10 species, they're all reduced compared to non-polluted conditions. So it hints that copper is actually stressful in these concentrations we used. And then we looked at the relationship between investment in sedimentary and the relative performance in copper polluted versus non-polluted conditions, meaning if the value is bigger than 1, they do better under copper stress, and if it's more negative, they do worse. You see that there is a positive relationship between investment in sedimentary and how well they cope with toxic copper stress. So then we did all these pairwise interactions. There were quite a few of them. So we actually completed them for a week, and we plated all the soil washers out and then counted colonies of the different color and morphology. And what we found is these are interaction networks. So these are all the 10 species, and they're basically labeled according to their investment in sedimentary production. So D is a really high producer, and A doesn't produce anything. And these are the pairwise interactions between these species. So the arches go from, at the top, it goes from left to right, and at the bottom, it goes from right to left. And what you can see is, under control conditions, most interactions are negative, meaning that in co-culture, the species perform worse compared to monoculture. So negative interactions are light blue. But as soon as you add copper, there's a shift in this in these interaction networks, with more positive interaction actually occurring. I don't know. Can someone mute their phone microphone, please? Yeah. Okay, cool. Right. So then if we then actually look at the relationship between sedimentary and how well they do in co-culture. So on the y-axis, we've got the log density in co-culture versus monoculture. If this is bigger than 1, there is actually a positive interaction. And if it's smaller than 1, there is a negative interaction. So species do worse in culture compared to in isolation. And you see here, again, these are all ranked according to their sedimentary production. So the rubbish ones are actually in purple, and the ones that do really well are in yellow. You see that there's a shift in the intercept in copper, with the more sensitive species actually benefiting from the presence of others. But overall, there is a negative relationship, which means that actually the ones that do produce sedimentary are more negatively affected by another species. But this effect is not different between the control and the copper. So, oh God, sorry. Now this thing has disappeared. So we found over very short timescales, more like a physiological response, we see that actually the genotypes that invest in sedimentary do better when they grow in toxic copper stress. But actually, there's also some sharing of sedimentary between different species. But this does not necessarily say what will happen over more longer ecological and evolutionary timescales. So what you could imagine is if there is toxic copper, this would select for species that actually are able to produce sedimentary because they are able to survive under these conditions. But it could also be is that species actually adapt and evolve the ability to become resistant. So if you then look at the trait variation in communities compared to what you see for an individual species, of course, if you've got more species, you've got a wider trait distribution compared to within a single species. So what we hypothesized is that actually selection should act by actually something called ecological species sorting. So selection should favor taxa that actually are able to detoxify copper rather than evolution within single species, because there's simply more variation within a community context compared to within species. So to test that, we set up evolution experiments where we've got one focal species, Pseudomonas fluorescens in green, and we evolved that one in copper and non-polluted compost in the presence and absence of the community. And we did the same for the community, also evolving it in isolation. So without the presence of the focal species. What we did do is we looked at changes in community composition, but we also did sedimentary assays, and we also looked at growth assays in the end to look at direct and indirect fitness as benefits underlying our observed changes. So if you look at the community, you can see here that copper in orange is actually favoring higher investment in Sadarfa production compared to the control. And this is because, sorry, I don't want to rush into this. So this is basically Sadarfa production within the community. This is the ancestral one. This is after 6 weeks of evolution. I forgot to mention it. And this is largely due to differences, changes in community composition. So all the treatments had a different, they initially started up with the same kind of community, but after 6 weeks of evolution, they differed significantly in their community composition. And this is due to actually copper selecting for taxa that have on average a higher Sadarfa production. So you can see here the control conditions and the copper. And here you see the 10 most common compost isolates. In red, it's the relatively high producers. In yellow, it's the low producers. And you see that there's a clear shift towards taxa that on average produce more Sadarfos. And this is the same if you actually add the focal species in the community, and also when you add copper and the focal species in the community. Sorry, it's not easy to go back, but more importantly, qualitatively, the patterns are the same where you have the focal species, yes or no. So copper selects against, favors high Sadarfa producers. God, where's this thing gone? Okay, so now if we look, this doesn't say anything about whether this is due to ecological change or evolution within the constituent community members. So it could be that Stenotrophomonas actually evolved the ability to produce less or more Sadarfos in response to copper-imposed selection. So to be able to pick up evolutionary change, we need to actually be able to track changes within a focal species. So that's why we actually embedded SBW25 in the community. And it's marked with a LAC-Z marker, so we were able to actually pick it out at the end of the evolution experiment. And what you find there is actually, again, in the no community and community conditions, pattern changes in Sadarfa production are qualitatively the same, but in this case, copper selects against really high producers. And that's in contrast to what we actually found in the resident compost community. So initially we were wondering, so why are there apparently opposing selection pressures at a community level and then within species level? So to actually look at that, we actually first plotted the distribution in Sadarfa production. So on the left, you can see the ancestral Sadarfa production, and you can see that our focal species, Pseudomonas fluorescens, has relatively high Sadarfa production compared to the community. But once you actually evolve it in copper, those distributions start to become looking more similar. So there is actually selection against Sadarfa extremes. So there's selection against really high and really low producers. And we look at individual genera in the compost, we find a similar pattern going on. So this raises the question, so why is there selection against low producers? Well, I think that is kind of obvious. If you don't produce any Sadarfos and you've got spatial structure in compost, then you might just simply not be able to survive and you're outcompeted by other community members. But why is there selection against really high Sadarfa production? So this could be due to direct and indirect fitness effects. So with direct effects, it just, what I mean is that basically the high producers simply produce too much Sadarfos, they don't need it to detoxify the environment. But there could also be like social exploitation between community members selected against really high producers if it benefits individuals that grow in the near vicinity. So to look at that, what we did is for all the evolved clones, we first wanted to know actually whether variation in Sadarfa production actually makes them more or less resistant to copper stress when they're grown on their own. So we can actually look at the direct fitness effects of variation in Sadarfa production. So all the evolved clones, what we did is we grew them in toxic copper sulfate, and then we actually measured their optical density over time. And then we compared the evolved with copper, without copper clones, and basically plotted those data, and we found nothing. So there's a clear response, phenotypic response, so there's clear variation in Sadarfa production, but it doesn't make the strains more resistant against copper. And what we then find if we plot the evolved clones from the copper treatment in red and the control treatment in blue, we indeed see that there's actually no relationship between investment in ZR production and copper resistance, meaning that actually if you produce more ZR4s, you are not more resistant, and that actually is costly. I'm not going into the details, but we also did some exploitation experiments between species and within species, and we looked at the role of iron in actually changing these patterns in ZR4 production. But what we found is that actually selection is largely results from these direct fitness, directly incurred costs. But I'm happy to talk about that into detail more later if you're interested in that. So to summarize all this work is like we found both evolutionary and ecological responses to copper pollution, and it results in trait convergence, so the selection against very low and very high producers, and this is due to ecological selection via changes in the community composition, but also adaptive evolution within species. So this is still, it's more realistic compared to what we do in media. But still, we don't know actually do these patterns hold in nature. So what we did is we went sampling. This is my only field work of the year, basically. Went sampling, we went to Paradise Valley, and we collected 100 soil samples. What we did is we actually quantified, together with the Camborne School of Mines, the metal concentrations in these soils, and we actually determined for 100 clones in a community of the soil samples whether they are ZR producers, yes or no. By doing colorific assays. So if you then look at that relationship, you find if you've got on average more heavy metals, you've got a higher proportion of ZR producers. So it would seem that again, in nature, if you've got more heavy metals, you favor ecological taxa that produce on average more ZR4s, or you've got a higher proportion of ZR producers in your sample. So it is important to know how these microbes respond to metals, because actually mine waste poses a great threat to ecosystem health and human health. And this is not going to go away over the next couple of years, because there will be an increased demand in rare metals in particular to actually support the development of more clean energies. So what people, and maybe Laura can correct me if I'm wrong, but what people often do now is actually remediate these metal contaminated soils chemically or physically by actually removing them from site. But there's more and more interest in actually using microbes to clean up mine waste. So that's maybe not surprising, because microbes are metabolically very diverse and they've evolved different ways to actually deal with heavy metals. And one of them is the production of ZR4s. So what we wanted to know is actually, does this natural remediation trade off with if you've got any chemical remediation? So we went back to the mining site, the former mining site, and we took 30 samples in this case. And what we did is we incubated them with lime or not with lime, with the idea that lime is alkaline, and that actually increases the pH, and that should precipitate the metals, making them less available to microbes. So, and as such, we would expect there to be selection against ZR production. Well, if we incubated those communities just as they are, we would actually expect there to be the maintenance of bacterial taxa that produce ZR4s, because only these are able to detoxify the environment. So first, we confirmed indeed that liming, which is depicted in red, which is a bit confusing because it's less metals compared to the control. So in this case, the control is more metals. And what you can see is actually, indeed, if you add lime, the pH increases. So that means that, in theory, this should result in a precipitation of some metals. And that's what we find if we look at the non-ferrous metals, so the non-iron metals. We find, in particular for copper, zinc, and manganese, I think, that if you add lime, these become less available, biologically available. And then if you then look at what the phenotypic response is in terms of ZR production, you see that in the liming treatment, there is there's a reduced number of ZR producers. And Also, the average Sadarephore production of the community is reduced, meaning that natural, so our remediation strategies, they select against natural remediation in these communities. And why is this important? Well, that's what I want to actually look at also in my fellowship. So I'm very precariously standing between two electricity cables. So Sadarephores, they don't only affect how microbes interact with one another, they also affect how bacteria and plants interact with one another. So some work has shown that if you've got like an individual bacteria that produces Sadarephores, if you plonk that in soil with a hyperaccumulating plant, then actually that plant will take up the metals more readily compared to in the absence of that bacteria. So in theory, if we could manipulate the community-wide level of Sadarephore production in communities, this might actually affect how microbes interact with plants and how many metals plants will take up. And that is important because there's more and more talk about actually using plants combined with metals to actually clean up mine waste in a more sustainable way. What I would like to do in the future is look at a division of labor between species. So I mentioned that different bacteria produce different Sadarephores. What I didn't mention is that they actually differ a lot in their affinity to different metals. So some species will be much better at binding to copper, while other species will have Sadarephores that are much better at binding zinc. So in theory, if we've got more metals than one in the environment, then actually species could specialize in detoxifying just the one metal and help each other out when there's more than one metal available. And actually, typically in these mining sites, there is contamination with multiple metals. And that's a bit about science, but I also wanted to actually tell you a bit more about actually what I've done in the ESI as part of the Creative Exchange program. It's been a while ago, I think 5-6 years, when there was a bid to actually be involved in one of these collaborations between artists and scientists. At that point, it was not specific to the Falmouth Exeter University campus. It could be any artist that you would like to collaborate with. So I got chatting to Paweł Sierzyński, and he's actually had a friend, a ceramicist, who would potentially be interested in doing one of these collaborations, because she said actually a lot of these glazes in ceramics, they're quite sensitive to metals, and that really affects the quality and color of the glazes. So what we did together is come up with where's the mouse gone? With a project to look at the effect of metal contamination on the quality of soil glazes. So she used my collection of soils that I quantified in terms of their heavy metalness, so the different metals that were in there, and we picked up some that were very varied. And what she did, that she burned the organic material and then used those to actually make glazes. And you can see this is before it's fired, and that's after it's fired. And then, although from this distance, don't look that different, there was a clear range in the different colors and the quality of the glazes. And in the year after, I went to an exhibition at the ESI, at the Eden project, and I bumped into Tim Cockrell from Falmouth Uni. It was an exhibition about like microbes and how cool they are when you really zoom into them. So we got chatting. I was like, it would be really nice to actually work together, because I've got all these really cool colonies, bacterial colonies, and they all look very different. So it would be nice to have to work together on that. And then I actually plated out some of these colonies to actually pick the 10 different clones that I used for the competition experiment. And what I found is that of the 46, there was one that was actually really swarming across the plate, but it was not swarming randomly. It was swarming towards certain other community members. And we did 16S sequencing on these clones, and it turned out to be a Penibacillus species. And that species is well known for forming these really intricate swirling patterns. But based on the observations I did, I was like, what if we actually can pair up different strains with Bacillus and then see how it actually would affect how these two different species swarm together over a plate. So can they use biotic and abiotic cues in the environment to actually determine where they will go on a plate? And we can. So on the left, you can see Variovorax, which is a quite common soil bacterium. And if you mix it with two different Bacillus strains, one that does produce sadarophores and one that doesn't, then the way they swarm over a plate is completely different. So in this case, the Bacillus is able to produce sadarophores, and it means that Variovorax is actually able to swarm together over a plate. Well, if you mix it with a Bacillus that does not produce sadarophores, this is not happening at all. And I really enjoyed these collaborations. So as part of my fellowship, I actually wrote down a bit to actually include some more of these art science collaborations. So I've just done a few together with non-profit organization Then Try This. So we had 3 different art science residentials, one at Wheel Martin, one at Soul Farm, and one at Fish Factory. And basically the title was Social Microbes, and the artists could hand in a proposal on anything tangentially related to social microbes. So we had some really interesting one about looking at the role of microbes and research on microbes in autism, one about how microbes interact in soil in terms of sustainable agriculture, no digging practices, and another one looking at how microbes inhabiting former mining areas, like in Wheel Martin, where they actually were mining for clay, how that could affect interactions. And these were all very interactive, so there was always a workshop or something else involved, so actually the wider public could engage. And with that, I'd like to thank everyone who helped out. It was a fair bit of plating and other work in the lab, and I'd like to thank you for your attention and coming along.
Have time for questions.
It doesn't even, wait, I can't stop sharing, I guess. Yeah.
Do you want me to do it?
Yeah, okay. And then I need to go, well, yeah, I'll do this if you.
Okay.
Yeah.
Any questions?
You know what, I can go back to this, so then you can see both the screen and me. Not sure if that's a good thing, but yeah.
Okay, I'll start with a question then. There's no question.
Dave.
Okay, sorry, go ahead. Okay, I have a question.
Yeah.
Actually, do you see within species, do you see any variation in siderophore production as a function of whether the species around are mutualist or competitor species?
Yes, because we find it within the focal species. So even if you add the compost community or not, you see a lot of variation. So there's the evolution of cheats that are actually whites that don't produce the main siderophore piverdin. But qualitatively, those patterns are quite the same, suggesting that there's actually limited exploitation between different species, but that actually interactions, social interactions, mainly take place within species. And I've got some follow-up experiments that actually show that. So there is exploitation within species. So if you produce fewer sadarophores, you benefit from a producer's presence. And this is negatively frequency dependent. That's what you expect with social exploitation to happen.
But then you also get that plasticity in production as a function of who's around you.
Well, plasticity is more regulation to me. So there is a lot of variation. I presume we can't measure that in soil. So we need to take the microbes into the lab and then measure that siderophore production in a common garden environment, in this case, ion limited medium. So we look at the possibility of producing siderophores and not at plasticity. But it would be really cool to actually do some transcriptomics on those traits and actually see whether there is that plasticity going on. Yeah. Dave.
Thanks, Elze. My question is about the applications for remediation of metalliferous land. And it just feels like a lot of remediation practices might traditionally have been to try to lock away metals,
Yeah.
make them not bioavailable.
Yes.
And in your system, you're talking about ways in which bacteria make metals bioavailable, and I can see why that might be good if you can harvest them.
Yeah.
But it feels like a fundamental change in how we might go about acting on that land.
I think it really depends on your ultimate goal. So I think if sites are really heavily contaminated, I don't think you can't use metal accumulating plants to get rid of the metals. I think that is just not realistic. So in that scenario, it would actually be better to lock away the metals by, for example, making them reside within the rhizosphere of plants rather than actually them being taken up. In that way, they don't end up in the waterways. So it's just that then it's more about stabilizing metals rather than actually making them more or less mobile and actually removing them.
Hi there, can you hear me? A question from the internet.
We can hear you.
Yes.
Hi.
Wait.
Perfect. Hi. Great talk. I listen with great interest. I do study mining impact sediments or catchment rivers, all this stuff. And then it was quite interesting when you did the experiment and you put the lime as well with the bacteria. And I wonder if you did a control with just lime and then lime and bacteria, because lime is used sometimes in just for control of decrease of pH, so that the metal they can precipitate.
I didn't do that because initially we were really interested in how actually it affects the microbial community. But I could imagine that there will be an interaction between adding lime and there being microbes there. Although liming did select against bacteria that make metals more mobile, so I don't see how you would interpret the data different than there being selection against detoxification in the community. And also, I don't think it would be realistic to actually have a treatment without any microbes, given that microbes are everywhere.
Everywhere, yes,
Yeah.
indeed, yeah.
Thank
Thank
you.
you.
Should I look at the chat?
Wait, there's one red dot.
Elze, can I ask another one?
Yes,
about
of course.
vegetables?
Yeah.
I might have asked you this
Yeah.
before.
Yeah.
You know I like to know, of course.
Who doesn't? Yeah.
How broad are they in their metal targeting mechanisms? So you talked about iron here and
Copper.
copper.
Yeah.
Yeah, but I mean, do they span across all metalliferous elements?
I think there's a lot of divalent metals, so it's like chromium, cadmium, zinc, nickel, has been shown manganese. There are therefore that bind a lot of different metals, but for individual species, usually the affinity is highest to iron and then one other metal. And we've done some, Luc and Ruth have done some experiments where they actually tested this for a model community consisting of 5 species. So for those 5 species, we know that some that are good in binding copper are not so good in binding zinc and the other way around. So it seems that there's not one general therefore that can bind all the different metals. So there's quite a lot of specificity going on.
I think that's something to do with
Yeah.
the structure.
The structure. For a lot of these, we have no idea what their chemical structure is, even for the ones that are quite well studied. Yes, David.
So in your experiments, or throughout all this, you've basically shown that external stress, in this case pollution, can lead to evolution of cooperation within communities, or species sorting within communities.
Yes, I wouldn't call this necessarily cooperation. I think I haven't shown it, so I only looked at community-wide functions. I think you really need to look at pairwise interactions, see whether actually one species benefits the other species, and actually that function is done partly because it benefits another species. So in this case, I would call it facilitation. It helps another, but because we don't know the origin of that function, it's hard to tell that it's actually co-evolution.
Okay, and this sort of facilitation then.
Yeah.
is that due to the unique properties of siderophores, basically there being a public good solution, or is that something that's more generic to external stresses that can facilitate the sort of?
So I think there's two things going on here. I think copper is reducing overall population densities, meaning that competition for resources is reduced. So that already makes the interaction sign less negative. But then also in some scenarios, some of the species do produce something that protects the others, and that's an external public good that is actually released into the environment. And by doing so, it makes the environment more habitable, better to inhabit, can't pronounce that, compared to when that sensitive species would be growing on its own.
Okay, so basically this isn't necessarily something that we can now make a generic statement of, saying that external stresses will always lead to cooperative or facilitative behavior.
I don't think you can. It really depends on the function you're looking at, I think. But it will reduce competition if it reduces population sizes. So there's less competitive, the interactions will be less competitive, I think.
Okay, thanks.
Yeah.
Hi, thanks, Elze. My question is actually about your art science
Yes.
collaboration.
Mhm.
And I'm curious if you feel they've impacted how you see this work at all, like how you see the communities you're studying, or how they allow you to think through any issues you've been dealing with.
So with the one with the big pictures of the microbes, it was clear on the plate that they were going for something, but it was really not possible with our current setup to actually look what was going on when you look at more detailed patterns. So that has really opened my eyes, and I really want to pursue that line of research in the future. So that was actually really beneficial to actually have that different approach to look at the same question.
Oh.
Yeah.
Any more questions?
Question on the chat.
Okay, not a question from Faye, just to say I love listening to this and really appreciate the synthesis of cutting edge microbial science with local environmental concerns and local artists. Actually, if time, what do you see as the most promising application of this research? Excellent.
I think it's answering a fundamental question. It's like understanding why different and under what conditions different species actually cooperate or not. But also, it would be great if we could actually use that understanding to come up with more development, more sustainable mining strategies. But I think that is still quite far away. So initially, it is answering a fundamental question with applied relevance, but it's not something I could patent anytime soon. I'm quite sure.
Always try.
Yeah, I just have a question about interspecies cooperation and the potential for it in this context.
Yes.
So you mentioned that in order to think of its cooperation, you need to have some sense that there's selection acting to facilitate on one species to facilitate the growth of another species. Why would that ever happen? And I mean, are there in this? Okay, so let's just rephrase that question slightly. Is there some sense that the species that aren't producing siderophores that might be being helped by the siderophore producing species are actually going to be helping the siderophore producing species at all? So in some way?
If anything, I think we find exploitation rather than cooperation in our system. So the helper species, so the ones that are facilitating the growth of the sensitive species in general do a little bit worse, but the extent of that effect was not that big. And I think that is because we've got spatial structure in our system. I reckon if we would mix it more, we would find more exploitation. So I think the only reason or possibility for cooperation would be if there's specialization in the division of labor. So one species does one thing and the other does another, and that actually in the end benefits one another.
I guess what could be the possibilities of that other? So you've got siderophore production. Are you saying different types of siderophore?
Yes, If one species produces siderophore A that can bind copper, another species produces B that can bind zinc, then that might end up being a mutualism. Yeah.
Great. Well, thanks so much, Elze, for a great talk. And thanks everybody for coming. And I think our next state-of-the-art is going to be end of January, and that's going to be with Joni Willett. So please do come along as well.